discuss the new topic. The name of new topic is the direct method of gene transfer. You know very well there are two methods, direct or indirect. Today we are going to discuss the direct methods of gene transfer. These are also known as vectorless method. Vectorless method, because in these methods there is no use of vector. That's why they are called vectorless. So there are so many methods. Out of so many, today we are going to discuss the four methods. The first one, which is given here, that is called micro injection method. Second one, which is called gene gun methods. Out of three, we will going to discuss electroporation method. Electroporation method. The fourth one, chemical mediated gene transfer. Here chemical methods. Chemical mediated gene transfer method. So first of all, we will start to discuss micro injection method, which is very important, which is given in your NCERT book. As the name indicates, micro means very small. What will we take here? We will take the syringe here. With the help of syringe, it will suck the foreign DNA. This foreign DNA, which is present within this micro injection syringe and needle, it is directly injected into the animal cell. This is called animal cell. This is called host cell. This is called nucleus. This portion is called cytoplasm. These are called pipette, which is called micropipettes, holding pipette, which can hold this animal cell. So this is the direct method. With the help of a syringe and needle, we can send the desired piece of DNA, which is called a foreign DNA, into your host cell. So this method is called micro injection method. The second method, which is more important, that is called gene gun method. Gene gun method. As the name indicates, this device, it is look like as a gun. Or with the help of this gun-like device, we can send the desired piece of DNA, and you can say gene of interest, into the host cell, which is called target cell. This is called gene gun method. This method was invented in 1987. In 1987 by a professor. The name of professor is called Stanford. Professor Stanford and co-worker in Cornell University. Cornell University of USA in 1987 here. So this is the most important methods. This method, in its method, Stanford and co-worker, they made a device which is called gun. In gun, what they will loaded? They loaded 1 2 micrometer gold and tungsten particle. These gold and tungsten particle, they are coated by the foreign DNA. Look, this is called a DNA that is loaded, and they can be suited. These gold and tungsten particle on the target target cell. This target cell is called a plant cell. Plant cell with the high speed, with the high velocity. That's why the look of this device is look like as a gun. It is called a gene gun method. It is also known by some name. It is called biolistic method. Very, very important questions. Biolistic method. It is also called bombardment method. Bombardment methods. And the simple is called gun methods. So make the diagram of these. The thing are labeled. This is called pin, which is called firing pin. Here, what will be fill up? This is the gunpowder, which is called cartridge, and this region at where you can be put the DNA, which is coated. DNA, it is coated around the gold and tungsten particle. This is the vent, and this is called stopping place. And from here, the target pair, it can be sent the foreign DNA, desired DNA for the target cell, which is called plant cell. So this is the main way by which we can be sent the gene gun methods, which was discovered by the Professor Stanford in 1987 in Cornell University of USA. The third one method is called electroporation method. Electroporation method. As the name indicates, when electric field is applied on a cell, what will be happen? The force which is present within this cell, here you can say cell wall, that is going to be increased. Porosity nature of plasma membrane. Plasma membrane is increased. The size of pores is increased. As a result, more and more foreign DNA it is taken by the cell from outside to inside. So this method is called electroporation, which work under the influence of electric field. The fourth one, which is called chemical mediated method. Chemical mediated method. As the name indicates, with the help of some chemical, the particular foreign DNA, you can say gene of interest, it can be sent into the host cell. Here, there are so many chemicals, out of so many chemicals which are given here. The one most important is called PEG. Very, very important questions. PEG stands for polyethylene glycol. Polyethylene glycol. This is the chemical. Another which can be used as a calcium chloride. The second chemical is called calcium chloride. Here you can say calcium ion, CaCl2, here you can say Ca2 positive. So with the help of these chemicals, we can transfer the particular gene into the host cell. So these are the four methods which are given. First one is called microinjection method. Second one is called gene gun method. It is more important. This kind of method of transfer of particular gene of a gene of interest into the target cell, and here the target cell is called plant cell. With the help of this device, which is look like as a gun, that's why this method is called gene gun method, ballistic method, microprojectile methods. These are the some names. This method it was invented by the Stanford. So this is more important. Next one is called electroporation. Increase the force nature of this plasma membrane so that it can be taken more and more DNA from outside to inside. The fourth one is called chemical mediated. These all methods are known as direct method of gene transfer. Here you can say foreign DNA. Foreign DNA. Here there is no use of vector. That why they are called vectorless. Already we have discussed it. Means in some cases we use the vector. The vector which can be used in that cases is called plasmid, bacteriophage, plasmid,